Welcome everyone again to the Biophysical Techniques lectures. This is the third lecture on fluorescent labeling, FRET, and light sheet microscopy. We're going to hear today from Ben Sutcliffe. Just to remind you again, or to let you know if you haven't attended any of the earlier lectures, that we'll be using the Q&A system to ask questions of the speaker. So you can type your question in during the talk or at the end of the talk, and Ben will go through the questions at the end and try to address the comments that you have. So if I could hand over to Ben then.
Excellent. Okay, so hello everyone. I'm Ben from the LMB Light Microscopy Facility, and I work with Nick, John, Jerome and James. I'm going to cover three topics today. Fluorescent labeling in microscopy, briefly cover some FRET, and also tell you a bit about light sheet microscopy. So I'll start off with labeling for fluorescence microscopy. So why would you actually want to do this? Well, it provides massive sensitivity and specificity in your sample. So you can label, here you can see microtubule labeling in a muscle myocyte, and you wouldn't be able to see it with this detail if you didn't use a fluorescent label that targeted just that protein of interest. You can also use it to easily multiplex different colors together, so you can label different proteins within the cell and find out where they localize and how they interact with each other. So you can see the chromatin here in blue, and mitotic spindles in red, and then the kinetochore in green, and how they all come together during cell division. And here in this breast cancer section, you can see DAPI in blue, cytokeratin in green, vimentin in yellow, and the ER in red, and how they all come together in that tissue. And this allows you to study localization and interaction within cells. So how will you actually go about doing this? Well, the most common method is immunofluorescence, which people typically just abbreviate to IF. So this is called immunohistochemistry in tissues and immunocytochemistry in cultured cells. It requires an antibody against your protein of interest, which can be either monoclonal, so directed towards a single epitope, and this is very specific, or it can be a polyclonal, so it can detect multiple targets on the protein of interest. Fixation methods are also extremely important to determine antibody recognition. So if you fix and it happens to occlude the epitope that you're interested in, then you won't get very good labeling. Sometimes you need to permeabilize a tissue in order to be able to get to the target of interest, for instance, if it's intracellular. There are two main methods of antibody labeling. So there's direct labeling, where you have a primary antibody which is conjugated already to a fluorophore, and this directly binds to the protein of interest. And then there's indirect labeling, where you have an unlabeled primary antibody here in black, and then the blue secondary antibody, which recognizes the primary antibody. And this one can be labeled with either fluorophores or other types of tags. This gives you a boost in your fluorescence signal, because you get amplification, because there's more secondaries binding to the primary, whereas here you just have one primary binding to one epitope. So as mentioned, chemical fixation. So unless you're going to do live imaging, then the cells need to be ideally fixed to immobilize the molecules within them and maintain the cytoarchitecture. And it also permits access for antibodies to their target antigens. So there are different types of fixatives out there. There's organic solvents, and these can permeabilize cells and tissues. So examples are the alcohols and acetones. So they remove lipids and dehydrate the cells, so you can get your antibody to the epitopes of interest. They're bad because they tend to shrink the cell, so you don't end up with accurate spatial information. If you want to have good spatial information, then you can use crosslinkers, sort of the paraformaldehyde, glutaraldehyde, glyoxyl. type. They chemically interlink the cellular content, so they keep everything exactly where it was. Some do it more quickly than others. It just depends on what you're actually looking at. If you're looking at high-speed events, then people actually recommend using glyoxal because it fixes things much more rapidly than paraformaldehyde and glutaraldehyde. But you do need to permeabilize the cell if you want to look at intracellular components. So the most suitable fixation method will always depend on what you're trying to detect and where it is. And if you're buying commercial antibodies, the data sheets will give you information about which compatible fixation methods you can use. So what actually is an antibody? What does it look like? Well, a conventional antibody, such as this IgG here, is large. It's about 150 kilodaltons. And it's made using mammals, so rats, mice, rabbits, goats, donkeys, etc. The head portion is known as the Fab fragment. And this has the top of this here is the variable domain, and that has the antibody recognition site represented by a star here. If one of these conventional antibodies is too large, so if you're trying to get inside deeper tissues, then you can use just the Fab fragment, which is this section here that will be cleaved off this antibody. And you've got, again, the antibody recognition site. If that's too large again, you can use a single chain variable fragment, which has just got the antibody recognition site, or the antigen recognition site in it. Other ways of getting smaller antibodies into your epitopes of interest is to use nanobodies. So, well, a heavy chain antibody with its nanobody. The nanobody is the head domain here, which recognizes the antigen. And these are made in camels, and you can get them from sharks, but people tend to just use llamas, alpacas, and camels. And using a nanobody is very good for super resolution, as it's very, very small, so on the order of 2 nanometers, and it can bind to your protein of interest. So you're not adding size to your protein of interest. For super resolution, that's important. So what can you then do to label these primary antibodies? Well, you can use secondary antibodies, which have an organic dye coupled to them. So the traditional organic dyes were fluorescein and rhodamine. These had low stability, and they were pH sensitive, and they're prone to quenching and things like this. So modern dyes developed in the last 15 to 20 years are much, much better than this, and they're very, very robust. They're difficult to bleach, and they're very good for live imaging and really open up the doors to enable you to do large Z stacks without too much photo bleaching and things. And these are very small as well, so they're good for, if you directly conjugate one of these onto your antibody, you're not adding much size to it. Obviously, if you use a secondary antibody, you've got that huge 115 kilodalton beast with a small organic dye stuck on the end of it. So you still have to remember that that's involved if you're looking at smaller proteins. You can also use a quantum dot. These are small inorganic fluorescent nanoparticles, about 2 to 50 nanometers, depending on which one you choose. So which wavelength here, as you can see, the blue ones are the smallest, the red ones are the biggest. And also whether you've got any extra functionality coated on the surface of the quantum dot. So they can be conjugated to biomolecules and to determine specificity. So you can have streptavidin ones, or you can have ones that are coupled to antibodies. They're very bright and highly photostable, and they have a very large absorption range combined with narrow emission spectrum. And this is ideal for multiplexing. So you can excite all the quantum dots with one single wavelength, and then you can image the resulting emission signals very efficiently with different bandpass filters or using different detection ranges on a confocal microscope. They're electron dense, so you can also use them for correlative fluorescence and then electron microscopy, as you can see them in the electron micrograph. But when you functionalize them, they can increase in size. So they're not great for accessing small compartments. So if you wanted to get them inside vesicles or synapses, then it might be a bit problematic. And they can exhibit blinking and a high propensity to aggregation. So there's pros and cons to all labeling techniques, of course. So if you don't have an antibody, or you don't want to use an antibody, what you can use is if you don't have an antibody, you can use an epitope tag, which then you can use another highly purified antibody to target and then label your protein of interest. So you generate a vector with your target gene and then an epitope tag on that. And then you can label it using antibodies that are highly available from companies. So there's the flag tag, HA, V5 and MIC, and they're all very good at what they do. It just means that you can make your transgenic and have different tags on different transgenics in the same either cell line or same animal, and be able to label them with different antibodies to allow you to do multiplexed imaging. So if you want to label without an antibody, or you simply don't want to use an antibody, I'm going to show you some different approaches to label biomolecules without them. So the first would be to label the protein directly. So this typically is done on purified protein. So if you're not inside the cells, you can then add it back into the cell if you wanted to see where it goes. And they rely on reacting to functional groups within the protein, or within the target. And the common reactive groups are isocyanate derivatives, succinimide esters, and malamide activated fluorophores. They can all react to the functional groups in your protein, and they tag it with a fluorescence such as fluorescein or tetramethylrhodamine. When you do this, however, you run the risk of over-labeling your protein, which can lead to precipitation. So you need to optimize the conditions before you go ahead, before you run the actual final experiment. And those are used typically in cuvette-based fluorescence measurements, so you can see how much of your protein is there, and also in vitro reconstitution experiments. Well, they can be introduced, as I said, into live cells by microinjection, so you can see where they go. You can also label cellular compartments with great specificity with various different commercial products which are out there. So there's fluorescent ceramide analogues which label the Golgi. There's lysotracker for lysosomes, ER tracker for the endoplasmic reticulum, mitotracker for mitochondria. You've got dye, which allows you to label the plasma membrane, typically used for labeling the processes in neurons. And then tubulin tracker for looking at tubulin within the cell. We can also take advantage of high affinity natural interactions, and this can help you to boost a signal within a cell. So here you've got your protein of interest labeled with a primary antibody, and then the secondary antibody, instead of being conjugated to a fluorophore, it has this biotin fused to the end of the antibody, and this is then recognized by streptavidin. And here that streptavidin has lots of fluorophores on it, and this can give you a massive boost in signal intensity. You can also use Paxil derivatives, which recognize the microtubules, and the typical one of that is tubulin, which is silicon rhodamine tubulin in the far red, and that's very, very good for microtubules. You can label actin with phalloidin, and also you can recognize acetylcholine receptors using this alpha bungratoxin, which is high affinity for the it's a toxin that has high affinity to the acetylcholine receptors, and here you get good labeling of those in the cell. Another way to label without antibodies is to use a fluorescent protein conjugated to your protein of interest. So this could be GFP or RFP. So the first fluorescent protein described was GFP in 1962, and that was isolated from this from the jellyfish here, Aquaria victoria. And how you use these is you express them as a tag onto your protein of interest. So you have to do some transgenics here. So you isolate your protein code, and you add an epitope tag, similar to adding an epitope tag, but you add a fluorescent protein tag on the end, and then you add that construct back into the cells of interest, and you can see where it goes. Doing it that way is perhaps not ideal, because you've got your protein of interest tagged with a fluorescent protein intermingling with the wild type protein, which is in the cell. So you can't be sure whether your protein of interest is going to be either perturbed by this tag or not, or whether the function in the cell will be disrupted. So what you can do is you can now do CRISPR-Cas9 to tag the endogenous gene, and then see whether that actually is causing an effect, or whether you're just looking at the localization of that protein. And there's a plethora of different colors that you can choose from now, ranging from all the way from blues all the way out into the far reds. So there's many things to choose from to label with fluorescent proteins. The RFPs were taken out of a coral, here, the discoma. And then from there you get the far red RFPs from that. And then the near red fluorescent proteins are coming from bacterial phytochromes, which can be imaged using 2-photon microscopy, but they do need a cofactor for fluorescence. So the fluorescent proteins have this beta barrel structure which protects the fluorochrome, which is in the middle, which is responsible for the fluorescence. And it protects it from large pH changes within the cells, so they're pretty robust for live imaging, although they do bleach more readily than an organic dye. Along the lines of fluorescent proteins, we have optical highlighter fluorescent proteins. So these are photoactivatable, so they're initially dark. For photoactivatable GFP, for instance, it's initially dark, and you switch it on using a 4 or 5 laser, and then you can see where your protein goes within the cell. So here you can see in this fixed specimen, they've just photoactivated the word GFP into the sample. There's also red versions, so PA M cherry, is a photoactivatable M cherry. Next, we've got photoconvertible fluorescent proteins, and these are ones that you can shift the emission spectra from green to red. So good ones of those are CADE, EOS, and Dendra, and you just hit those again with a 4 or 5 laser, and then you can see where your red protein ends up. And then we've got photo-switchable fluorescent proteins, and the emission spectra here can be reversibly turned on and off by illumination with specific wavelengths of light. So you visualize your protein, you can turn it off, and then you can turn it back on again. So these can be used for doing sort of palm imaging, which is a form of super resolution. And there's also iris FP, iris fluorescent protein, which is photoconvertible and photo-switchable. So you can convert it from green to red, and then you can cycle it on and off. So there are ideal tools for investigating protein dynamics, and also palm. And John will give you more information about super resolution techniques on Thursday. You can also use proteins to track the age of a protein pool. So there's this timer, Tandem Fluorescent timer protein. So you've got 2 different proteins, one that's a slow maturing and one that's fast maturing, and you can look at the ratio between the 2 to work out the age of a protein pool. And here you can see in these 2 dividing yeast cells here, you've got a young, an old and a young spindle, and then they move to different cells. So you can see track which is the daughter cell and another cell. There's also cell cycle labeling. So you can have a protein which changes its fluorescence emission depending on which stage in the cell cycle you are. So you can track cell cycle progression in live cells. And that can be proved to be very useful, because you can see whether any things you've done to the the cells are causing them to stick in or stay into G1 phase, S phase, or G2, for instance. And then you can do quantification of that automatically with different softwares. We can also use a type of fluorescent protein called mini-SOG for correlative fluorescence and EM. And it's mini-SOG stands for mini singlet oxygen generator. It's a 15.5 kilodalton fluorescent flavor protein, and it comes from Arabidopsis, the plant. So when you illuminate mini-SOG, you generate singlet oxygen, which can catalyze local polymerization of diaminobenzidine, or DAB. And this is an insoluble osmophilic reaction product that you can see when you look at it by EM, because it becomes electron dense over on top of what you see in the normal tissue. So it allows you to use strong aldehyde fixation, which preserves the ultrastructure, but would hinder the access to antibodies and gold particles. And it also doesn't require any permeabilization. And it works in mammalian cells, intact nematodes, and Drosophila and rodents as well. So here you can see an example of the fluorescent signal, and then also then looking in the EM, the same magnification. Oh no, sorry, this is just light microscopy at the electron or the diaminobenzene signal, and then here you can go in with EM and look at that signal at much higher resolution. So the next technique is to genetically fuse a non-fluorescent peptide or protein that then binds to a fluorescent ligand. So there are different types of these. There's self-labeling and enzyme-mediated. So I'm going to go through the self-labeling versions of these. So for Vitrogen, you can get flash and reash. So these are membrane permeable derivatives of fluorescein. So the flash and resorphin, which is the red version, or reash. And they're initially non-fluorescent, but become fluorescent when they bind to a recombinant protein containing your tetracysteine tag, so this tag here. So you can have your protein with the tag on it, and when you add flash or reash, you don't see anything until it binds to the protein of interest. So this is good because it means you don't get lots of background in the media, so because these don't fluoresce until they're actually bound to the protein of interest. However, you can get non-specific labeling of thiol-rich biomolecules, so molecules that have high amounts of cysteine in them. So it's a good idea to check whether this is going to happen before you go ahead and do the experiment. Another version of this, which is highly specific, is to use chemical labeling. So you can have your protein of interest, and then you can add a small snap, clip, or halo tag onto those. And these will then react covalently with the tag. So you have the ligand. So here you've got benzylguanine, here benzylcytosine, and here a chloroalkane. And they covalently react with the tag and link a label onto the tag. And this is a very rapid process. So you can get rapid labeling in the entirety of, for instance, a Drosophila brain in 10 minutes versus doing a long 2-day antibody protocol. So it can be very quick for doing screening, for instance. And now with CRISPR and Cas9, you could tag any endogenous protein in the cell and then label it in your animal, and label it with one of these tags, which are readily available from these companies, NEB and Promega. And you can also get custom ones from Genelia Farm, from Luke Lavis's lab. So these are some examples of what you can do in a Drosophila brain. So this is the picture that's behind my head, which has got showing you multiplex labeling in 2 subsets of neurons in the brain. And then here we've got some chemical rainbow in the brain, so you can use it to tease apart the connections and the tracks within a complex expression pattern in the brain. There's also enzyme-mediated labeling, so these require you to use an enzyme. They don't happen without the enzyme. So these are ACP and MCP tags. They're much smaller than the snap, clip, and halo tags at 9 kilodaltons, but they do require an additional enzyme to catalyze the labeling. So if you can't get the enzyme into the tissue, then you also can't get the labeling to happen either. So they are non-cell permeable, so therefore they're only suitable for labeling surface proteins, or you can permeabilize and label inside a cell. And their specificity is determined by the enzyme that you use. So the ACP synthase will modify predominantly the ACP tag, whereas the SFP synthase will modify both the ACP tag and the MCP tag. Therefore, you have to do a sequential incubation if you require dual color labeling for this. So another way to label without antibodies is to do this bioorthogonal labeling. So this has been developed at the LMB by Jason Chin's group. I'll just briefly tell you about that. So it works by the introduction of an unnatural amino acid into your target protein, which can then be labeled with a fluorophore. So in order for this to happen, you need to engineer the sequence of your protein of interest so that it contains a UAG codon in the mRNA. And this will be at the site where you want to add the tag. You then express an orthogonal amino acyl tRNA synthase and tRNA pair, and this can then add the RNA to that UAG. And this can either be transfected as a plasmid, or it can be by creating a transgenic animal. And then you feed the unnatural amino acid to the cell or organism, and the orthogonal synthetase and tRNA pair will now be able to incorporate that unnatural amino acid into your protein. And you have engineered the UAG site to be in the right place, and then therefore you're hijacking the natural transcriptional apparatus of the cell to sort of tag your protein. It can be labeled by reaction between the unnatural amino acid and a tetrazine linker attached to a quench fluorophore, so you can, without labeling, you don't get any fluorescence, and then when it binds to the unnatural amino acid, you can see the fluorescence. And this can be very useful for tracking protein expression within the cell, because it's tightly regulated by the machinery within the cell, and also the availability of the amino acid you supply. So just to go over and summarize this part of the talk. So I've shown you that you can do multiplexing with fluorescent labeling to label multiple different proteins within the cell, so you can see how they are interacting with each other. It provides you with specificity and sensitivity in fixed samples. That's the main reason why you want to label something. And so you can see it above and stand out from all the other things that are in the cell. If you're just doing wide field, bright field microscopy, it'd be very difficult to pinpoint a particular protein in the cell. The chosen fixation method is very important. So people tend to just do 4% paraformaldehyde, but it's not always the best way to go, particularly if you have fast-moving proteins which you want to freeze in a particular moment in time. There are also other techniques available that allow you to do live cell imaging with high specificity, particularly the fluorescent proteins most commonly used. And you can also do rapid chemical labeling in thick tissue. So next, I'm going to give you a bit of information about why you might want to use Förster resonance energy transfer, or typically called FRET, but some people also call it fluorescence resonance energy transfer. So the diffraction limit of confocal microscopy is a little over 200 nanometers. It allows you to see sort of subcellular organelles and things like that. Resolving anything smaller than that is a bit tricky, and John is going to give you an insight into how you might do that with super resolution on Thursday. If you want to image sort of intra or intermolecular interactions, sort of at the single molecule level, such as looking at protein folding upon ligand binding or protein-protein interactions. So these are things that typically happen below 10 nanometers and therefore are well below the diffraction limit. So you can't really resolve these using standard microscopy or super resolution techniques, but you can if you use something called FRET. So it's a readout for these intramolecular and intermolecular interactions. So it was first described in the 1960s by Förster. That's what gives it its name. But what it actually is well, the key thing you need to know is that it's the transfer of energy between two dipoles, known as dipole-dipole coupling. So when the donor molecule here absorbs a photon, it gets to the excited state, and then through coupling of its dipole to the acceptor dipole, it can transfer the energy rather than emitting a signal. It can transfer, or emitting a photon, transfer the energy to the acceptor, which then emits a photon. And this is what FRET is. There are various requirements for FRET to work. First of all, you need to have two fluorophores, a donor here, CFP, and an acceptor. So this one here is YFP, well, Venus, a particular YFP. And they have to meet several conditions. So in order for FRET to happen, you have to have overlap between the emission spectra of your donor and the excitation spectra of your acceptor. So if they don't overlap, you won't get FRET. You also need to have them close enough together. So if they're further apart than 10 nanometers, then you won't see any FRET occurring. And they also need to be in the right orientation. So the dipoles need to be lining up and parallel to each other. If they're not, then you won't get any FRET. So if these conditions aren't met, then you won't get any FRET. So the classical fluorophore pair is CFP and YFP, or CFP and Venus. Venus is a new version of YFP that lots of groups are using. So here's the fluorescence excitation spectra of CFP. And you would optimally excite this using a 436 nanometer laser, if you have one. So this here is the emission spectra of CFP. And for doing FRET, you would detect this signal using a 460 to 500 nanometer bandpass filter, or the same detection window of a confocal detector. Here's the excitation and emission spectra of YFP, so shifted down towards the red end of the spectrum compared to the CFP. And when you overlay these, you can see that the peak excitation of CFP, you're not going to get much excitation of YFP, a very, very tiny amount, but it's negligible. And here, where the emission spectra of CFP overlaps with the YFP excitation spectra, is where you'll get your FRET happening. So there's a big overlap here between CFP and YFP, so indicating there'll be a good FRET pair. So there's a strong spatial dependency in FRET, and it's determining what determines the FRET efficiency. So this is simply the number of quanta transferred from the donor to the acceptor divided by the number of quanta absorbed by the donor. And the biggest influence on your FRET efficiency is this inverse to power 6 relationship between the separation distance of the donor and acceptor molecules, so this R number here, and the Förster resonance, the Förster distance, sorry, of the donor and acceptor pair, so the R0. And this is the distance at which energy transfer is 50%. So when R equals R0, FRET is 50% efficient. For an example of this using the Psi3 and Psi5 donor acceptor pair, when they are 4.9 nanometers apart, you get FRET efficiency of 50%. If they were to move closer together, then you would get an increase in FRET efficiency. If they would move further apart, then you'll get a decrease in FRET efficiency. So you can use this to calculate the distance between your donor and acceptor, and therefore you can use FRET as a molecular ruler. So you can look at molecular interactions that are less than 10 nanometers. So here's a Jablonski diagram showing you what's happening inside the fluorophore. So when you excite the fluorophore, you get, with the right excitation wavelength, you get a photon driving an electron up into the excited state. And then from here, you can look at various different things in FRET. You can look at the behavior of the donor in the presence of the acceptor. You can look at the acceptor behavior upon FRET. You can look at the lifetime. So this is the acceptor behavior upon FRET and the behavior of the donor in the presence of the acceptor. You can look at the lifetime of the donor in the presence of the acceptor, and you can also look at photobleaching rates as well. So in terms of the methods that you can use FRET for, I'm going to go through these today, just to give you a bit of an overview on what you can do. So acceptor photobleaching is the easiest way to obtain FRET measurements, and it's the most straightforward. So the principle is that in the absence of the acceptor, no FRET can occur. Therefore, there's a resulting increase in the emission from the donor. So the signal from your donor molecule should be increased. So following photobleaching of the acceptor, the donor is no longer transferring any energy to that acceptor and can use all that energy itself to relax back down to the ground state and emit photons and fluoresce. You can also use this with a non-fluorescent acceptor, which can be chemically destroyed to give you increased donor fluorescence. So here's an example in some fixed cells. This is before you bleach your, so this is the donor and this is the acceptor. So before you bleach the YFP, you've got strong YFP signal here and a sort of average CFP signal from both of these two cells. When you bleach that top cell, you bleach the YFP, you get an increase in the fluorescence of your CFP in that same cell. And here's a control cell below. So this indicates that you were getting FRET before. You can also do sensitized emission, and this looks at the acceptor fluorescence when you excite the donor, and is presented as a ratio of the FRET signal to the donor signal. This is the most commonly used form of FRET, as it can be done cheaply with a wide-field microscope with just 3 filter cubes, one for the donor emission, one for the acceptor emission, and one for the donor excited acceptor emission. And since this is fast, it can be done with live cells, so you don't have to use fixed samples for this. But you do have to have plenty of controls to control for crosstalk and things like that. I will go into that in a moment. So these are the artifacts that you can have in the sample. So the crosstalk issues. So this problem of crosstalk is due to the fact that we're working with 2 fluorophores which have overlapping spectra, which is what we need for FRET to work, but it can be a bit of a problem when it comes to giving crosstalk. So here, if you excite the CFP donor with 405, 440, and 458 nanometer wavelengths, you can see you get varying excitations of your Venus as well. So this is called excitation crosstalk. So if you were to use 440, you're still going to be getting some Venus excitation. So it would be best here to try and use 405, I guess, just to limit that. Even worse is the emission crosstalk. So the emission spectra is overlap much more. So when you detect the peak signal for Venus, which is here, there's a strong chance you're going to also be imaging your CFP because you're still getting 40 to 50% peak emission of your CFP. So you need to do all of the correct controls to correct for these eventualities and to calculate your FRET donor ratio. It's necessary to do a number of image processing steps. One way to simplify your approach to doing sensitized emission FRET is to use spectral imaging and then mixing. So this is where you detect the whole spectrum at once. And we have 4 systems at the LMB which allow people to do this. So they all have this multi-channel photon multiplier, which is a bank of, well, you image the light to a confocal pinhole onto a diffraction grating, which is then spreading out the spectrally dispersed light over a 32-channel array of detectors, each of those collecting 9 nanometer of the visual spectrum. So, and they're adjacent to each other. So you can detect 288 nanometers of the visual spectrum. And because you do that all simultaneously, you can scan the entire spectrum very quickly you can do live imaging this way. And then as long as you have all the controls, so the donor on its own, the acceptor on its own, and a background sample as minimum, and you can unmix these. So these are the different spectra for the controls, and then you can unmix everything that you get back on the detector to give you your autofluorescence, your CFP, and your YFP, so the FRET. So it can be very useful to use this to make your FRET experiments a bit easier. You can also do fluorescence lifetime imaging with FRET. So usually when you excite a fluorophore, it's a very quick process, and it stays in the excited state for a couple of nanoseconds before it bounces back down. So here is up in the excited state before it bounces back down into the ground state and emits a photon. So in the presence of an acceptor, this lifetime of the donor is changed. It's also changed with fluctuations in the local environment, such as pH and other ions. So here's an example of that. So if we have GFP molecule and we measure the lifetime of its emission signal, we can plot the decay for that lifetime, the green curve up here. And you do this with a pulsed light source and a detector which measures after each pulse how quickly the signal arrives at the detector. You do that for a long, long time, and you end up with a type of decay curve that you get here in green. In the presence of the acceptor, in this case M-cherry, which is fused to GFP, you can see that the reduction in lifetime here indicates FRET, so between these two. And just by getting the decay times in the presence and absence of the acceptor, you'll be able to calculate the FRET. This requires, however, a special system which actually is able to measure as fast as you want things to be measured. A word of warning is this can work nicely if done correctly. However, you can seriously reduce your FRET efficiency if you mount your fixed samples in the wrong type of mounting media. So here you can see that live cells work fine, fixed cells seem to be fine, but then if you mount them in Prolon Gold or Fluorogel, you get a massive drop in the FRET efficiency, so it's not ideal. One final thing that you can do with FRET is you can combine it with fluorescent proteins which are attached to molecules to measure something within your cell. So the first one published was the yellow chameleon, which is a chimera of CFP, the calcium binding protein calmodulin, the calmodulin binding domain of myosin light chain kinase, M13 here, and then YFP. So when you add calcium to this, you get a conformational change in the protein which brings YFP and CFP close together, and you get a high FRET signal between the two. And when you remove calcium, you go back to a non-FRET state, and you can also then use trypsin to a low FRET state, and you can use trypsin to break the calmodulin, and then these two separate, and you get no FRET, so you can use that as a control. So this was used by Bill Schaefer when he was in Robert Tian's lab to look at the calcium transients in the pharynx of a C. elegans as it's chewing. So again, it's just chomping away, and you can visualize that with this FRET sensor. So these are all the controls that you should do and recommended for the different techniques that I told you about today. And this talk's going to be available, so if you do need to do these, rather than scribbling it all down now, you can take a look at the talk later. But there are many controls that you need to do to be guaranteed that your FRET is true. And it's also a good idea to do more than one approach just to confirm that the FRET you saw in one technique was actually the correct result. So in summary, you can use FRET to measure interactions below the diffraction limit. You can also use that, use it as a molecular ruler. It's useful as a biosensor, but you must do all your controls, otherwise there's no point spending all the time doing the FRET in the first place if you don't do the correct controls. So now I'm going to tell you a little bit about the light sheet microscopy possibilities that we have at the LMB. So this is our light sheet, sort of plethora of light sheets has increased recently after we've been doing a bit more development in them. But first of all, why would you want to use a light sheet? I mean, mainly it's for live imaging. Typical ways of imaging, as I'll demonstrate in a moment, give you high phototoxicity and bleaching, whereas with light sheet you can get gentle, fast imaging and therefore image your sample without it dying. So, however, there's no one light sheet that fits all samples, so you can't just buy one system. You have to have a system tailored around your microscope. So these are all the different types of light sheet that are out there. So you can't just buy one and hope that it's going to do everything you need. The reason is that it's usually designed around a particular sample that you want to image. So you have to illuminate it in a particular way and mount it in a particular way. So you end up with lots of different types of light sheet as a result. So initially it was a new type of microscopy, but now it's considered just a new type of illumination technique that can be adapted to the model system that you're imaging, and it explains why there's so many different acronyms. So you can't just put, I used a light sheet microscope in your paper, you have to sort of say which type it was. So why is it better for live samples? So if you look at a wide field versus a confocal microscope here, you've got excitation of the entire specimen in this wide field over here, whereas you're just looking on a single focal plane, imaging that onto a camera. So it's very quick, but you're getting out of focus excitation above and below the focal plane, which is not ideal. So that gives you a fuzzier image. So to improve your optical sectioning, you can use a confocal. So you just focus your beam down to a single point on the focal plane, and then you scan that backwards and forwards to build up the image in X, Y, and then scan it through in Z. So here you're also getting excitation above and below the focal plane. You're just not seeing that because you have a pinhole, which prevents it from getting into the detector. So that gives you the optical sectioning, but you're still getting the photo bleaching and phototoxicity over time. So why is a light sheet better? Well, you're just imaging the same area that you're illuminating. So your sheet just illuminates along the focal plane, and that focal plane is imaged onto the camera. And then you can then move your sample through the sheet, so nothing else moves, just the sample. And you'll only get any bleaching or phototoxicity along the plane of focus, not through the entire specimen. So it's much gentler on the live sample. You can also vary the thickness of your sheet depending on how much you fill the back aperture of the illumination objective. So these are your illumination objectives here. If you underfill them, you'll end up with a thicker but longer sheet. So that allows you to look at a thicker sample, so you get more of it focused in the field of view. If you only have a tiny sample, so if you're looking at a Drosophila embryo or even just a few cells suspended in agarose, you can fill the back aperture of your illumination objective to the maximum, and then you'll get a thinner but shorter sheet, so you can only look at smaller objects then. So this is the microscope that I'm currently developing. We did previously have an M-spin, which was similar in design, sort of grew out of an open spin, but it needed some improvements. So it's now will have interleaved two-sided illumination, so it switches from one side to the other. It gives you more even illumination because you don't have both sides of your sample being illuminated at the same time when the images are taken, and then you have to with this system, you'll have to fuse the images later. This gives you fewer artifacts. It's also going to have axial scanning to give you a thinner light sheet. So it's using an electrically tunable lens here to move the sheet through the sample. So similar to a confocal scan, I guess, but it means you can have a thinner sheet, which is a bit shorter, and then just quickly scan it across the sample with the same frame rate, the same rate as the rolling shutter on the camera. It has 4 laser lines, so you can do multiplexing. It's temperature controlled, and it's going to be used for organoids, spheroids, and Drosophila embryos, things of that sort of size. Anything up to about 500 microns should be okay in this sample. And the construction is almost complete. And it's got a theoretical resolution. This needs to be measured when it's finished for GFP of 255 nanometers in XY and 475 nanometers in Z. So this is how I would mount the sample in here. So we've got, this is an embryo, Drosophila embryo in a column of agarose. You can also do different things that would, some small hooks that you can hook onto something, or you can rest something in this little cup, which is mounted on a syringe. And you can also mount things in FEP tube and suspend them in the sheet. So these are some examples of from the previous system. So things should be better than this, but we can definitely get single cell resolution from these Drosophila embryos. Here's a mouse oocyte that was imaged very early on when the Shoe Lab were in the building. And then we can also do techniques that allow us to overcome scatter. So on a light sheet microscope, well, not all light sheets, this particular type, you have the ability to rotate your sample through the sheet and image it from different sides. So it's like holding an apple up above your face, and you can rotate it and look at it from different sides to get a full picture of it. So you can get, you can optically sort of, well, not optically, you can computationally fuse these images together to give you a nice 3D reconstruction of your image. It's not as trivial as that. You need to have beads within the sample to do segmentation in Fiji, or you can do it using image intensity profiling in Huygens, commercial software from SPI. It does work though. So here you can see Drosophila salivary glands and the sea of beads that are used as fiducial markers. And you can image those over time and do a, so you can see the salivary gland growing, and then you can also do a 3D render of that. So it does work, it just takes a bit of effort to get there. And we can also do, this is using just image intensity profiling. So this is a kidney organoid from JP in the local lab. So not only do we have that we'll have that light sheet microscope, we've also got a field synthesis light sheet, which was built by James Manton. So this is very, very similar to the lattice light sheet developed by Eric Betzig, only much, much simpler. And it's best for cells on a cover slip. So here, these are dictyostelium, extremely fussy beasts to image. They just like to die when you try and look at them. But here, James has managed to image them for over 10 minutes, and they just happily go around doing their macropinocytosis as happy as if it was in a dark room. And this is a sample from Rob Kay, and he's really happy with this microscope. And James is also now developing an OPM in the building. So this is sort of a video and a schematic from Andrew York, who came up with this idea and sort of is building them as well. But he wants to make it open source and everything, so James is building one as well. This should give you the same resolution as the field synthesis light sheet, but you're only using one objective lens effectively. You don't have to illuminate with illumination objectives and then detect with a detection objective. You illuminate and detect with the same objective. And the advantage of this is that you're going to be able to put any sample carrier, so any typical sample carrier that you already use, so slides, imaging dishes, chamber slides, multi-roll plates, but not quite yet. on top of this detection illumination objective and then image them with a light sheet. So it's going to be very rapid and also gentle, the imaging. So it's not yet built, but James is working on it and it should happen soon. I'll let this video finish so you can just see everything. Obviously it's just a schematic. It's not what the actual thing will look like. But it's going to be very fast and high speed. It's also going to have temperature control, so you can do live imaging on it, which is the whole point of doing light sheet. Really, it's gentle live imaging of your sample. The problem is mounting it. So the fact that you can put any sample carrier on there is great. So in summary for this part, so light sheet allows you to do live imaging on much longer time periods with less photo bleaching. You can look at larger samples much faster than with other methods. And with Multiview, you can get more isotopic resolution. And we're going to have 3 different systems in the building to take lots of different types of sample. And you can also do clearing to allow deeper tissue imaging, but then this is not going to be a live sample. They're only applicable for fixed samples. So thanks for your attention, and if you've got any questions, I'll have a look in the Q&A and see what we came up with. Thank you.
Okay, thank you very much, Ben. So do we have any questions? Yeah, okay. I'll leave that to you, Ben.
Hi, Nina. So Nina is asking when the OPM will be available, and if so, will it have CO2 control? It will have CO2 control. Yeah, a full incubation system will be on there. I'm not sure when James is planning to have it ready. Before Christmas, he said the end of January, but he didn't specify a year. But I'm hoping it's going to be soon because he's not that far off now. But we will let everybody know when it's ready. Tiana's asking a specific question about the OPM as well. Will it fit an alico? And what clearing agents are suitable? So in terms of the XY size of an alico, yes, it will fit it, but I don't know whether if you're typically, because you typically do those with sitting them on a little mesh, which is about 1 millimeter from the cover slip. That's the problem. So you've got that working distance to overcome. The best thing to do would be to have a chat with James about that and see if you can overcome it. Regarding clearing agents, you can use any type of clearing agent you want because you just put your sample on top and it doesn't touch the lens. There's no dipping like you would have on the M-spin, which I'm developing, which you can't really do much clearing because you don't want to put the nasty solvents into the sample chamber and melt your lenses. Laura Whitworth's asking, will there be capacity for CL2 samples on the OPM when it's operational, or any of the light sheet microscopes? That'll be a conversation that you have to have with us and Nick and Chris Tate in the building. I guess as long as we can ensure that nothing will leak out, it would be fine on the OPM, I'd imagine. It'd be difficult on the other two purely because of the way you mount your sample. You have to dip it into a bath of solution and then image it that way. So it'd be hard for those to be CL2 safe. But with the OPM, I should imagine it would be okay. But that would be a conversation you'd have to take up with Nick and everyone else. Marcus is asking, could you comment on the labeling efficiencies I presented and how suitable it makes them for threat studies? I'm not sure if I understand your question. Could you comment on the labeling efficiencies of the labeling strategies you presented? Well,
I think maybe Marcus is asking about the specificity of where you think the label is and how efficient it is, or the efficiency in
Well,
getting?
yes, that's.
I guess.
I mean, if you're going to do FRET, you're going to have to use a fluorescent protein tagged to your protein of interest. So you want the epitope that you tag it to be external to the protein. You wouldn't want it to be inside, really, because then it wouldn't be able to get close enough to the acceptor molecule. Yeah, so it all depends on where you put the protein, where you put your fluorescent protein on the protein of interest as to how good the FRET's going to be. And also, FRET will tell you how efficient it is. If you've put them in places which are get close to each other when the two molecules come together, then you'll get good FRET. If you put them on places in the proteins which are distal, when the proteins bind to each other, then you won't get any FRET. But the way to determine that would be is if there's any other biochemical studies of the proteins that allow you to know the orientation that they're in when they bind to each other, and in which case you can then choose epitopes that will give you, or tag sites which will give you good FRET when they do bind to each other. And Viola's asking, so Marcus has followed that up. I assume that for FRET you want every molecule labeled, but depending on your labeling strategy, not every molecule might be labeled. Yes, that's true. So if you want every molecule labeled, then you'd have to do CRISPR or Cas9 within live cells in order to label all of the endogenous proteins of a particular type. And then you'll be able to see them. You know that two proteins coming together will give you FRET. For the others, I guess you'd have to do other techniques in order to determine how much of your labeled protein there is versus the wild type protein, to be able to give you an idea of how efficient your FRET will be. So Viola's asking, would it be possible to use the light sheet for autopsies in brain after paraffin removal? I don't think the light sheets that you might, depends how big they are, but you might be able to do that on an ultra microscope. So they're designed for bigger samples. Light sheet. It's a light sheet that's designed for bigger samples. And the LMB, we're focused on smaller things. So the brain organoids are kind of the biggest thing that would go onto any of these light sheet microscopes, and they can be up to 1 millimeter across. In which case you'd be able to image those on the OPM to some extent, but you wouldn't get those into the MSPIM or the field synthesis light sheet. Okay, well, I hope that answers the questions.
Okay, yeah. Well, thanks for a great talk, and also thanks for some great questions.
Yeah, thanks everyone.
And we'll see
you. That people listened.
We'll see you all on Thursday for the final episode of Light Microscopy, where we go below the diffraction limit with John's super resolution talk.
Thanks everyone.